In this section of Unit 1, we will continue our journey of looking within a single cell and explore how different components of the cell are truly interconnected to allow the cell to survive and thrive. Specifically, we will be exploring how proteins, or the doers of the cell because they do everything, are made from transcribing DNA into mRNA and then translating that mRNA into functional proteins. These processes are what allow life to persist, and they are constantly taking place within your cells too, ensuring that your body continues to function so you are able to complete, well, this lesson, right? So thank you, cells! So let's start by honing in on just a brief example overview of organelles working together. Organelles within a eukaryotic cell must all work together in order for a cell to survive and thrive in their environment. A really good example that displays organelle cooperation is the process of protein synthesis. Remember that synthesis just means the formation of something. So protein synthesis, the formation of proteins. It all starts in the nucleus, where DNA resides. The instructions to create a particular protein are found in a gene in the DNA of a cell. So as to prevent damage to this DNA, though, when a protein is required to be synthesized, a copy of that particular gene is encoded onto an mRNA, or messenger RNA molecule. This process of transcribing the directions that are found in DNA into a small segment of mRNA is called transcription. The mRNA molecule then leaves the nucleus through a nuclear pore. The mRNA molecule then travels to a ribosome, so we went from nucleus to ribosome. This ribosome can be located either in the rough endoplasmic reticulum or be free-floating in the cytoplasm. Remembering back to our other material that we learned about, right? These are where we find our ribosomes. So there in the ribosome, the mRNA sequence is then translated into a sequence of amino acids to form a polypeptide chain. This process is appropriately called translation. Remember now, poly means many, and peptide refers to the long chain of amino acids that are all held together by peptide bonds. So polypeptide basically is another way of saying protein or the precursor to a protein. The next stage is called post-translational modification. This is where the polypeptide chain is modified to create the final protein. This may be done in the rough endoplasmic reticulum or in the Golgi apparatus. Now, being directly exposed to the water-containing, what we call aqueous conditions of the cytosol has the potential to alter the newly developed protein's shape, right? And as we know, shape is directly related to function in proteins. So this can cause the protein to become non-functional, right? Thinking back to our hydrophobic exclusion from Unit 0. Therefore, when a protein leaves the rough endoplasmic reticulum, it does so in vesicles. And these vesicles are pinched off from the endoplasmic reticulum and then confused with other organelles, such as the Golgi apparatus. In the final stage of protein synthesis, the newly created protein is transported around the cell, again in vesicles, to wherever it needs to be for it to carry out its function, right? If it's helping out as an enzyme in the citric acid cycle, then it'll be delivered to the mitochondria. If it's a channel in the plasma membrane, then it will be delivered to the plasma membrane where it will be a transmembrane protein that'll create a channel for other molecules to pass through. So in that final stage, those newly created proteins are transported all around the cell in vesicles, but sometimes the vesicles may fuse with the plasma membrane and secrete the proteins into that extracellular space, so that way the proteins can carry out their potential functions in that space or move on to other cells where they can carry out functions in other cells. So this is just a really brief overview of organelles working together in a cell. Now, seeing how we apply all of our knowledge surrounding what organelles do and seeing how they all come together to carry out their function.